Good evening, everyone. Welcome to Science at the Theater, Secrets of the Soil, sponsored by the Friends of Berkeley Lab. My name is Jeff Miller, and I'm Head of Public Affairs. This is our final Science at the Theater event for 2011, but please don't be sad. We'll be back in the spring, although there is some talk that we might do something special during the winter, and if you want to know about that, the best way to find out is to join the Friends of Berkeley Lab. There's some information about it at the front desk, and you can also sign up at our website. One of the many wonderful features of working at the lab is to be surprised, enthralled, and yes, sometimes humbled by an emerging field of science. Tonight's topic is a perfect example. Who would have thought that the story of soil would contain a cast of characters to rival a Tolstoy novel? Indeed, the story is so rich, and the climate and energy ground we have to cover tonight so vast that we needed 5 scientists to even scratch the surface. Leading tonight's team is our moderator, John Harte, who's a professor of ecosystem science at Berkeley, a Guggenheim Fellow, and an author of, 190 scientific publications, including 8 books. One of his books, Consider a Spherical Cow, is a widely used textbook on environmental modeling. John's particular area of research focuses on climate change and biodiversity. Should I say that he's still a very down-to-earth guy? I guess I will. Okay. Before John takes charge of the evening, though, a couple of closing comments. Please note the fire exits. We don't expect any emergencies, but just in case, please make a note. As usual, we follow the scientists' presentations and discussion with a question and answer period. There are microphones here, and for those of you who are upstairs, there are also some on either of the flanks. Please remember to keep your questions specific and short, and we like to move things along, so if you would like to ask a second question, please go back to the end of the line. Lastly, if I may ask, who is here for the first time? Please raise your hands. Very good, some in the top level too. Please take time to fill out the response cards, the audience survey cards, and return them to a volunteer at the front when you exit. And with that, I will now introduce John Harte. Please welcome him with a warm round of applause.
Thanks very much. Oh, thank you very much for organizing this event, and thank you all for coming. I'm going to introduce the speakers in a row, and then they will get up and talk. But before that, I want to make a few comments. You're going to learn a lot about soil science tonight, but in my comments, what I want to do is try to put soil science into a larger context. When we look around our planet, from the Arctic tundra to the tropics, from rivers to the sea, from the forest to the desert, we see a myriad of splendid detail, a complex mosaic of soil and water, of living creatures arranged in fascinating patterns on the landscape, and of the chemicals and the climate that both nourish and sustain these creatures and these patterns, and in turn are shaped by them. The quote that I'm showing in the upper left on the slide is one of my favorites from the entire scientific literature. Charles Darwin expresses here his confidence that the intricate details of ecosystems will be shown someday to be governed by laws of nature, by universal theory, just as the phenomena of physics are. And so he states that it's so interesting to contemplate. He calls it an entangled bank, but that could be any ecosystem. And he says that if you consider the many kinds of plants and the birds, the insects, the worms crawling through the damp earth, it's interesting to reflect that these elaborately constructed forms, so different from each other and dependent on each other in so complex a manner, have all been produced by laws acting around us. The genius of Charles Darwin was reflected in his capacity to be both an astute observer of nature, fascinated by the myriad details, and a scientific visionary, capable of conceiving a unifying theory that made sense of all those details and could explain the long history of life on Earth. And so in this quote, Darwin has posed a challenge to ecologists to do for ecology what he did for evolution and the history of life on Earth. The challenge is to develop a predictive and comprehensive theory of ecology, a science of the biosphere. And such a science will have to encompass evolution, but evolution is not enough. And it will have to encompass soil science, because soils are inextricably linked, intertwined with everything that happens in nature. The search for this unifying theory of the biosphere is urgent. There's a rapidly increasing threat to our planet from global climate change, from deforestation, mismanaged land use, the spread of toxic substances, and much more. The situation demands of us scientists that we learn to predict the effects of global changes on habitats, on species, and then the harm to society that will result from damaged ecosystems. If the science of the biosphere is to consist, if it only consists of stories, of particular explanations, of particular phenomena, but doesn't lead to comprehensive, overarching theory with a predictive capacity comparable to that of, say, the hard sciences, then I fear that ecologists will not be taken seriously. Not the way, for example, physicists are when they testify about whether, let's say, an asteroid is going to hit the Earth. So as you listen to the presentations this evening, I'd like you to pay attention to the ways in which these scientists are working toward achieving that goal. We're not there yet. There's a long way to go, but we're making progress. But ask yourself, are we peering at odd tidbits of fascinating but disconnected detail? Or are we progressing toward an understanding of the secrets of soil that will help us achieve a predictive grand theory of the biosphere? Such a theory will help society develop a reliable strategy for charting a course to a more sustainable future in harmony with the natural splendors of the world. And with that, I want to introduce the speakers. I'll introduce them in turn from right to left, the order that they'll be speaking. And they will then get up and make their presentations. And at the end, as you were told, there'll be an opportunity for plenty of questions. Let me start with Margaret Torn on the right. Margaret is the head of the Climate and Carbon Sciences program at the Lawrence Berkeley National Lab. She studies the natural carbon cycle and human influences on the carbon cycle, which has a tremendous effect on climate and climate change. She studies this through looking at land use and climate change itself. Margaret received her PhD from ERG, the Energy and Resources Group at UC Berkeley, in 1994, and I'm proud to say that I was her graduate advisor at ERG. The next speaker will be Eoin Brodie. Eoin is a microbial ecologist. His major research interest lies in better understanding the factors that control microbial communities and what happens when these communities change. He received his PhD in microbial ecology from University College Dublin in Ireland in 2001. Trent Northen is our next speaker. He's a staff scientist at Lawrence Berkeley Lab's Life Sciences Division. His research aims to construct detailed models of the metabolism and the energetics of cellular communities. He came to the Berkeley Lab in 2008 receiving a PhD in biochemistry from Arizona State University in 2006. And our last speaker on the left will be Janet Jansson. Janet is a senior scientist at the Lawrence Berkeley Lab. Her research is in the area of molecular microbial ecology and genomics with a focus on soil and marine sediments. She received her PhD in microbial ecology from Michigan State University, and she came to the Lawrence Berkeley Lab in 2007. And with that, we'll turn to our first speaker, Margaret Torn.
Thank you, John. Hello. Okay, let's see. In 1837, President Franklin D. Roosevelt wrote, "The nation that destroys its soil destroys itself." And he wrote those words during the Great Depression, when erosion and loss of topsoil were making the land so infertile that crops were dying in the field and people were starving. Those conditions were immortalized in John Steinbeck's novel, The Grapes of Wrath, and also in the photos of Dorothea Lange and other photographers who documented the devastation. And I have one of those work project photos up on the screen showing a really desolate field that should have had wheat on it. President Roosevelt was thinking about soil as a provider of fertility, for food to eat, for fuel to burn, for fiber for clothes. So he said that as the nation's soils go, so goes the nation. Now, 70 years later, we find soils at the center of a very different environmental crisis. Because it turns out that as the world's soils go, so goes the climate. And in particular, the organic matter in soil has the potential to alter the path of climate change in this century. The pivotal role of soil in Earth's climate is due to its role in the global carbon cycle. So as you know, CO2 is the main anthropogenic greenhouse gas, and plants consume CO2. This is a little hard to see the red, so there's some carbon dioxide molecule. Plants consume CO2, make organic material. This is input to the soil when plants die or shed leaves. And then in the soil, that material is transformed by microorganisms, the bacteria and fungi that live in soil. And the microbes may also decompose that material all the way back to CO2, so creating a carbon cycle. And this process can take CO2 out of the atmosphere if the material is stable in the soil, but it can also pump CO2 back into the atmosphere if decomposition is accelerating. And to give a feel for the magnitude of those flows and those stocks globally, consider that the natural flow of carbon from soil and soil respirations, that's that microbial decomposition, is about 7 times bigger than all anthropogenic activities of emitting CO2, deforestation and fossil fuel consumption and cement manufacturing. And second, to give you a sense of the stocks, I made the soil box and the atmosphere box. Those are to scale. There's about 3 times as much carbon in soil as organic matter as there is in the atmosphere as CO2. So you can tell that even a small change, a transfer of carbon from soil to atmosphere or an acceleration of decomposition would greatly change the amount of CO2 in the atmosphere and thus the amount of climate forcing. So one of the most important things that soil do in terms of climate is just to store carbon that would otherwise be in the atmosphere as a greenhouse gas. So let's look at anthropogenic emissions, and I'm just going to use the last decade as an example. So between fossil fuel combustion and deforestation, humans emitted about 9 billion tons of carbon as CO2. And that's each year. But the amount of CO2 in the atmosphere each year only went up by about half of that. Here it says 47%. The rest of those emissions were actually taken up or removed from the atmosphere through exchanges with terrestrial ecosystems in the ocean. Now, in the ocean, that CO2 forms a weak acid, which can have environmental impacts, and that's a story for another Science in the Theater. But 1/2 of that sink, of that material that didn't stay in the atmosphere, is taken up by plants and is stored in wood and soil. Excuse me, let's see if I can go back to that. And that's a very valuable service. Because if we think of climate change as the cost of running our economy on fossil fuels, we're actually getting a 50% discount right now. We only have to deal with half the impact of our emissions because of that sink through natural processes. So I just want to take another look at the land sink. As you can tell, so plants and soil take up about 25% of our emissions, but the sink is very variable. It seems to track climate variability from year to year that's affecting new plant growth and soil storage, but we don't actually know the mechanisms of how climate affects this sink, and we don't know in detail where that carbon goes. So can we say with any confidence that this sink will continue, that it can keep pace with our accelerating emissions, until we understand what controls the sources and sinks of carbon, the flows in and the flows out? We can't say that, and we don't know whether we'll continue to get this discount on our emissions, but I'll note that all the climate projections you see assume that we do. So we're sort of being optimistic in that one realm, but we don't know if it's true. Now I want to turn to the other reason that we want to know about the mechanisms. How is it that carbon is stored and lost in soil? And that is because of the potential for soils to release carbon dioxide and form feedbacks to climate change. Now, there are many long-term experiments. I thought John Harte would mention his in the Colorado Rocky Mountains, which is warming soil. Here's another one close by at Jasper Ridge across Highway 280 from Stanford. And from these experiments, we're learning something about how soils respond to climate change. And just quite simply, the most important message for this talk is that when you warm the climate, you warm soils. That's pretty obvious. And what we know from the experiments is that when you warm soils, microbial activity increases. And the decomposition rate increases, and you put more CO2 into the atmosphere. So when it gets warmer, you get more soil respiration of CO2 into the atmosphere. Now, if you get more CO2 in the atmosphere, you get more warming. And if you get more warming, you'll get more microbial activity and more emissions. That's a positive feedback that is amplifying our initial perturbation of the climate. And when it comes to climate change, what we call positive feedbacks are usually a bad thing, in that this is amplifying the effect of our activities. I wanted to give you a sense of the magnitude. Okay, so soils can form these feedbacks, but how big could this effect be? According to the Intergovernmental Panel on Climate Change, a kind of middle-of-the-road projection of climate change is about 3 degrees Celsius warming by the end of the century. That's about 5 degrees Fahrenheit. And this is middle of the pack of projections, but it's a lot of warming in terms of its impact. But based on those ecosystem experiments, we have a rule of thumb for how warming affects soil respiration. So what I did is I plotted the global soil respiration flux versus a warming of 3 degrees, and you can see that there's a small increase in that over time. Now let's look at, this is showing the anthropogenic CO2 emissions over this century that would give you that 3 degrees of warming. But what happens if that warming really does cause this amount of increase? So here's what becomes the new CO2 emissions to the atmosphere, added to the atmosphere if it's our direct emissions plus that increase in emissions from the effect of warming on soil. And you can see that what looks like a rather small change in a natural flow, but because that flow is so big, it becomes a really huge effect on the future climate forcing. Now, for most scientists, that 3 degrees is kind of at the upper limit of dodging many climate catastrophes. And this soil feedback then could definitely push us over that limit. And that's why John spoke of the urgency of understanding soils. So an important question is, how accurate is this projection? I used a pretty rough rule of thumb to make this plot, because we don't really know how much carbon in soil is vulnerable and what actually will control the rate of release. And so there's a pressing need to know that. Will the response to climate change be larger or smaller than what I have plotted here? So in the past, our view of what controlled soil carbon was rather simple, and it said that soil carbon was controlled mainly by the chemistry of plant inputs and the recalcitrance of human substances. And you don't need to know about that, in part because what I'm saying now is that we know neither of those things are really that important. And that's in part thanks to research by people at Berkeley Lab. So the question remains, why does some soil organic carbon persist in soils for tens of thousands of years, while in other places or times it only lasts a day before being converted by microbes to CO2? So in my research and in the research of many people at Berkeley Lab, we're trying to investigate that question with field work and lab work and collaborating with scientists all over the world. And we're developing new views of soil carbon cycling that we hope will give us a more accurate picture. And in a nutshell, this emerging understanding is that we could view the persistence of soil organic matter as an ecosystem property. It depends on the plants, the climate, the parent material, the soil itself, the microbes, and the intricate physical, biological, and chemical interactions among all those things. But unfortunately, this more complicated ecosystem view doesn't give us a simple answer to how vulnerable soil carbon is, but it is putting us in a better position to investigate it. And for example, we have a really exciting new project in Alaska coming up, and Janet will talk about her work in Alaska, sites like these. Another thing this new view does is point to the centrality of microbes, the living beings that are actually decomposing the organic matter. All these processes are carried out by microorganisms. And if we knew what they will do and how they will respond, then we would actually have the answers to our questions. I want to frame the mystery of soils in the words of Leonardo da Vinci, not just a brilliant artist and engineer, but also somewhat of a natural scientist who conducted some pot experiments on soils in the early 1500s. And he wrote, "We know more about the movement of celestial bodies than about the soil underfoot." Now, in 1500, European societies did not know very much about celestial bodies and their movement. So this means they didn't know very much at all about soils. So now we know much more about celestial bodies, and I think there have been many great science in the theater talks about them. But the living soils actually still remain a mystery. And to close, I just wanted to give one comment on that. So here's a photo of celestial bodies, galaxies and so on. And astronomers tell us there are about 100 billion stars in our galaxy. Now this is a photo not of celestial bodies, but actually of microbes in soil that have been stained with a phosphorescent dye. And on the poster for this event, you saw a handful of soil. And the microbiologists tell us that in a handful of soil, there are about 100 billion cells, or individual microbes. So it's the same amount of stars in the galaxies as there are microbes in just 10 grams of soil. But one difference is that we cannot see these microbes in their native environment, no matter what telescope we make, what probe we have, and we don't know very much about them, and we certainly have to be much more clever to get at them. In fact, many fewer than 10% of these microbes have been cultured to see what there is. It's probably less than 1% have been described of all the different taxa. So we don't know how to predict what they'll do. So let me leave the mystery of soils in the hands of the people who best understand microbes, and the three speakers who follow me are all experts in microbial processes around the world. And first is Eoin Brodie. Thank you.
Thanks, Margaret. As John said, I'm a microbial ecologist, and what a microbial ecologist does basically is look at the interactions between microbes, and not just between microbes, but between microbes and their environment. And it's very important to study microbes in their natural environment, and that's what we try to do. What I'm going to talk about today is some of our work in tropical soils. I'm particularly interested in tropical soils because microbes in tropical soils are incredibly important in terms of the carbon cycling. Actually, microbes are important in all soils in terms of carbon cycling. But they have a potential to show a very large feedback to global climate change. Microbes in tropical soils can also potentially hold the key to our energy future, and I'll discuss a little bit about that. So we know tropical soils and tropical forests are incredibly important for biodiversity. About 50% of the Earth's plant and animal diversity are found in tropical regions. But they're also the most important carbon stores annually on Earth. This plot here, if you look at the green areas, this shows plant productivity. And if you look at the tropical regions, plant productivity is maintained consistently throughout the year in the tropical zones, while it varies quite significantly in more temperate and higher latitude regions. What this means is that these regions store more carbon. They're active more frequently during the year, so they store more CO2. And this makes tropical regions the largest carbon sink on Earth. However, climate predictions are predicting significant changes in precipitation. If you look at this map here, the tanned areas show where precipitation is predicted to decrease. And if you noted what I showed in the previous video, that a lot of these decreases are predicted to occur in climate regions, in tropical regions. Now, these regions are historically and have evolved under low variation in rainfall, but now this drought is potentially threatening these regions, and the organisms in these regions are not adapted to variations in rainfall. So this might have significant impacts on the carbon cycle. And the previous slides were models, so these are projections into the future. This shows actual data from 2000 to 2009. And what we can see here, if you can see the red areas, this shows decreases in plant productivity. So decreases in plants, most trees in this case, consuming CO2, pulling CO2 out of the atmosphere and acting as a carbon sink. This is occurring mostly in tropical regions, And this is resulting in a decrease in the magnitude of these sites as a sink. So where does this carbon go? Well, the reason for this carbon leaving the soil is microbial enzymes. And the same way that we use microbial enzymes in our biological detergent to remove ketchup stains or tomato sauce stains of 3-year-olds, this is my son, the microbes in soil take plant litter, leaves, roots, trees, break that down into smaller parts, and those smaller parts so that they consume and gain energy from these compounds. Some of this material goes into soil as organic matter, some of it stays in soil, and some of it is released to the atmosphere as CO2. And the magnitude of that CO2 release is controlled by, in some part, by the microbes, what types of microbes are present, and how they respond to changes in climate, such as changes in rainfall or changes in temperature. And we're very interested in understanding these processes from a microbial point of view. So we partnered with Wendy Silver and Tana Wood from UC Berkeley to see how microbes might change in response to a drought effect in a tropical forest soil. Wendy and Tana simulated a drought effect in this forest in Puerto Rico. And the reason we're working at this site in Puerto Rico is that there was a 10-year study looking at rates of plant litter decomposition across 28 different sites. And it was discovered that this site in Puerto Rico had one of the highest rates of plant decomposition of all of these sites that were studied. Another reason that we're interested in this site is that it's teeming with microbes. And Margaret mentioned there were 100 billion microbes in a handful of soil. At this site here, we find 100 billion microbes in 1 gram of soil. And that's about 100 to 1,000 times more bacteria in a gram of soil than you'll find in any fertile soil. So my group, Nick Bauskull and also with Kristen DeAngelis and Julian Fortney, went to Puerto Rico and worked along with Wendy and her group. So how do you study this range of diversity and an enormous amount of microbes in soil? Well, as Margaret mentioned, we can't use satellites, and microscopes are probably about the right scale to visualize microorganisms. But the problem is when you look at microorganisms under the microscope, this is pretty much the range of shapes and sizes that you see. So we have millions of types of microbes, but a very limited range of shapes. And what makes this problem all the more challenging, as Margaret said, we can only grow a small fraction of these organisms in the lab, so it makes it very difficult to study these. Well, thankfully, in the late 70s and early 80s, Carl Woese and his colleagues, they determined that you could classify and identify most of life on Earth by comparing the DNA sequence of a few common genes. If you look at this box here, this is the metazoans. That's where we are, and all animals. Land plants are here, and the rest of this diversity is microbial. So you can see microbial diversity dominates the planet. We tend to think that we're in control of things, but we're not really. In the early 90s, the Department of Energy initiated the Human Genome Project. And this was a multinational effort. Many billions of dollars went into this and culminated in the sequence of the human genome, which is 3 billion bases after about 10 years of work. This also spawned a new industry and new technologies. And now with the evolution of these technologies, we can now sequence the soil genome, or the genome of all the organisms in soil. That's about 300 times the size of the human genome. So the technology to enable this and the development of this technology has outpaced Moore's law, and it's been pretty outstanding. So we now have tools to be able to understand soil microbial communities, to be able to identify them, and to be able to at least have some insight into what role these microbes perform without needing to cultivate them in the laboratory. So we went to Puerto Rico with Wendy Silver. They constructed these rainout shelters. And these are effectively perspex screens that deflect some of the rainfall. So we're simulating a drought effect here, reducing the rainfall or the throughfall by about 40%. We take some soil samples at various depths in soil, and then we perform some experiments. Some low-tech, using canning jars for incubations. Some high-tech, sequencing the DNA of the microbes in the soil, measuring enzyme activities, and various parts and components of the carbon cycle. This generates enormous amounts of data, So we use the computing resources up the hill at Berkeley Lab and also at the Joint Genome Institute. So what have we found? This is an ongoing project, so I'll give you some examples of what we've found so far. In a half gram of soil, we detected more than 4,200 different types of bacteria, and that's a very conservative estimate. With rainfall exclusion, we lost about 15% of that bacterial diversity. But even with this loss of bacterial diversity, we could see that certain groups of organisms increased. And these are actinobacteria. Some of you may have heard of these. If not, you've all smelt them. These are the organisms that give soil the earthy smell, particularly pronounced after a rainfall event. They also produce a lot of antibiotics, but they're major players in soil carbon decomposition. So their increase is potentially very significant. We also identified that the enzymes that degrade plant polymers increased in activity following drought. And some of our work with Trent has shown that the soluble carbon compounds in soil, this is the product of microbial degradation, are very different following drought compared to control conditions. So together, this is telling us that the microbial communities have changed, the carbon cycle has changed. And this work is ongoing. We're following this up, and we'd like to see how long it takes for these microbes to return to the conditions before this drought event, and how long does it take the carbon cycle to reset. Working in places like this also allows us to develop a fundamental understanding of how microbes work. And we would like to be able to take that information not only to understand how microbes affect the carbon cycle, but use those microbes to engineer processes to maybe curb some of the effects of climate change. So the DOE Joint Bioenergy Institute in Emeryville is currently working on developing biofuels from cellulosic biomass. And cellulosic biomass is basically the non-edible parts of plants, the woody components, the parts of plants that give it structure. Now, one of the reasons for this is that this does not impinge on food production, and some of these plants, particularly grasses like miscanthus and switchgrass, can potentially be grown on marginal lands also. But how do you go from these woody plants, grasses, to a biofuel and to fueling your transportation? Well, it turns out that this point here is really the bottleneck, or one of the major bottlenecks, and that's breaking down these plants. These plants have evolved over time to avoid degradation, but there are microbes that have specialized in breaking down these polymers. We want to break down these polymers and release them as sugars to produce fuels. So what would you do if you were a microbe? Where would you hang out if you were a microbe that specialized in degrading plant material? Well, you got to think like a microbe. So if you're a homeowner, you don't like the look of these. These are termites. But if you're a microbe that specializes in degrading plant materials, this is one of the places to be. Also, the rumen of a cow, we know that these are extremely efficient at degrading plant materials, and they make a biofuel. It's methane. If we could harness some of that activity, that would be very useful. Or you could live in a tropical forest soil with some of the highest rates of degradation on Earth, of plant root degradation on Earth. So my colleague, Kristen DeAngelis, traveled to Puerto Rico and decided that she was going to go and capture plant-degrading bacteria. How do you do that? How do you capture bacteria? Well, you use bug traps. Just like you capture insects, just like you may capture mice, you use bait. In this case, the bait we use is plant polymers, lignin or cellulose or hemicellulose. And Kristen and her colleagues took these porous beads, coated them in lignin. You can see this here. This is a great place for microbes to live. You saw the video at the beginning. Microbes like these porous structures. So they can hang out in here. They can chomp away on plant polymers. You bury them in the soil. Come back a few weeks later. And then you analyze which microbes grew best on beads with lignin and which microbes grew best on beads without lignin. So we use our suite of tools, DNA sequencing, and this device, the PhyloChip, which we invented to rapidly identify microbes in samples, look at what enzymes are being produced. And Kristen was able to identify that the beads that were coated in lignin were enriched in actinobacteria. Again, these plant polymer degrading microorganisms. And she was able to show that higher lignin degrading activity was present on the beads that were soaked in lignin. So where does this information go? Well, obviously, these microorganisms have enzymes capable of breaking down lignin, and this could potentially be used in biofuels. So these organisms and their genetic information is then passed on to other people working at the Joint Bioenergy Institute to more effectively produce enzymes to degrade plant material and hopefully reduce one of the bottlenecks to efficiency in biofuel production. So what I hope you can see is that it's important to understand microbes in their natural systems. These microbes have evolved in nature, and nature has allowed these microbes to become specialists in particular functions. If we can take advantage and understand the distribution of these microbes in nature, then maybe we can understand how microbes will respond to climate change, how they will affect the carbon cycle with their responses, and potentially how we can use the properties of these microorganisms to mitigate some of the impacts of climate change. Thanks.
Okay, well, it's a pleasure to get to talk to you. And I'm going to be talking a little bit about a different type of ecosystem. So Margaret told us how important soil microbes play in climate change and in stabilizing soil and sequestering it from the atmosphere. And then Eoin told us about tropical soils and their role in cycling carbon. I'm going to shift and talk about these more desolate regions of the Earth, these arid regions where plants don't do so well. And we might think that life doesn't really exist there. They're one of the largest areas of Earth. These arid regions make up something like 40% of the Earth's terrestrial surface. And if you're like me, you like to go there, and while you're there, you're mostly looking at the scenery. You're looking at the beautiful canyons and valleys and seemingly endless landscapes. And you don't look down beneath your feet. But underneath your feet is a whole nother landscape. And this is a landscape that's created by microorganisms. And so what we're seeing here are little canyons in the surface of the soil. And these are places where plants don't live very well because there's just not enough rainfall. There's not enough moisture in these soils. But microbes have adapted to live there. So if we could zoom in and look even closer into this soil, we would see these real pioneers, these cyanobacteria. So this is Microcoleus vaginatus, a very old species. The oxygen that we're breathing right now came during a great oxygenation event that happened by cyanobacteria. They got their metabolism going. The atmosphere was full of carbon dioxide, very low in oxygen. These guys started photosynthesizing and released oxygen billions and billions of years ago. And they're real pioneers. They're able to do things completely by themselves. They can take a little bit of dust from the air, a little bit of moisture, nitrogen from the atmosphere, carbon dioxide, and make everything that they need. So I find these absolutely fascinating organisms. And these are major inhabitants of the deserts, if you could only see them beneath your feet. And so here's a picture I took a couple weeks, or it was taken of me and one of the fellows in the lab a couple weeks ago out in northern Arizona. And these are what's called desert biological soil crusts. And I actually brought some samples today. So after this is all over, I'll run up there and you can come see them if you like. These can cover up to 70% of the total surface in, like for example, in the Colorado Plateau, in Arizona and Utah. But they're not plants. These are actually photosynthetic microbes. So they're absorbing the light from the sun, taking whatever moisture they can come by, a little bit of nutrients from the dust, and they're photosynthesizing. And they have their productivity as if you had spread a leaf across the whole desert. So they're very important in these ecosystems. And they've been around for a long time. The oldest fossil records, or some of the oldest fossils, are stromatolites, which are these sort of filamentous cyanobacteria binding to soil particles that are as old as 3.5 billion years old. So in comparison, vascular plants have been around for about 0.4 billion years. So we might speculate that there was a long period of time when these crusts ruled the world, and plants had to figure out a way to get up above the crusts. And so this is a picture comparing fossilized crusts with the modern crusts, and just looking at how their morphologies are very similar. Okay. So these are regions, and if you visited them, these are the sort of life at the limit of what you can survive in. And very rarely rains, less than 25 centimeters of water a year. And I'm going to tell you a quick little story. Eoin and I and a couple colleagues went out to collect some of these crusts. And one of the beautiful things about them is when they're dry, they're not doing anything. They are completely shut down. I'll tell you a little bit more about that in a second. And so if you collect them when they're dry, you can bring them back to the laboratory and you can wake them up. And basically, no time has passed for those guys as long as they're dry. And so we go out to the desert to collect these samples. And what happens? As it rains. And so here we are, madly trying to collect our samples before the rain comes. And it was off in the distance, and we kind of heard some thunder, and we said, there's no way that rain cloud is going to come towards us. And sure enough, it keeps moving closer and closer. But we managed to get everything packed up, and then, pretty soon it dries out. And so how would you live if most of the time you're dry? Metabolism does not happen when there's no water. We need the water for the metabolism. And so these crusts are comprised of these organisms that can kind of turn their metabolism, turn their living, active life on and off, just as a function of whenever they're wet. And there's many organisms that can turn on and off, but it's some sort of developmental process, like making a seed or something like that. And that takes a long time. These guys may be wet for a couple hours or a day, and then all of a sudden they dry out and they have to stop. So that's a very amazing capability. I'm going to show you a little video of these guys waking up right here in this panel. So this is dry sand. I don't know if we could dim the lights a little bit. You might see it better. Oh, there you go. You can see a little oxygen starting to come up here. Yeah. And these guys, they live underneath the sand, and as soon as you wet them, they, even though they're microbes, have the ability to move up onto the surface. And so you'll see it start getting greener and greener as they move along. And so this is one of their adaptations, is that they hide underneath the sand so they can use the sand as sunscreen to protect them from the UV damage. And then as soon as they get moistened, they slide back up onto the top of the surface. And this phenomena of being able to stopping metabolism was first observed by the original microbiologist, Leeuwenhoek. So he found these things in his rain gutter he called anomolecules. And he'd take the dirt out and he'd put it underneath, one of the first microscopes, and he'd look at it. And when he would wet the dirt, all of a sudden these little things would come to life. Was this spontaneous generation? Was this life happening, coming from nowhere? And that raised many theological concerns for a long time. But what that actually was were these little rotifers that could stop, basically desiccate, and then when they're wetted, they could restart their metabolism. So that's, and when we think of metabolism, that is thousands of reactions. So one of my particular interests in studying these desert crusts is how do organisms basically turn off? Our lives, we live it as a continuum. You're born and you live all the way through. But what would it be like if, one day it rains and you're on and you're living your life, and then the next day it dries out, and then you don't know what it's going to be like the next time you wake up. Is it going to be the winter? Is it going to be dark? Is it going to be incredibly hot? And so these organisms have to be prepared for anything. Okay, so these crusts stabilize the soil. The reason why they're called crusts is that they're very firm. So this is what a piece of crust looks like. And this is an actual result of the microbes themselves. So this is that Microcoleus vaginatus I showed you. And it's all these little beautiful little necklaces of cells that wrap themselves in this sponge, an exopolysaccharide that can hold that moisture longer, right? So they keep them wet a little bit longer. But this polysaccharide also sticks to the soil particles. So you can see it's all wrapped around the soil. And it holds the soil together. And I was just talking to a scientist in Washington last week, and I told him I'm interested in Microcoleus vaginatus. And he said, "You mean the organism that's holding down the West?" Because these crusts are what really stabilize the soil. And as Eoin mentioned, climate change is a real concern in these arid regions because these are already regions that have very low rainfall. These are organisms kind of at the brink. And as you change the seasonality, when it rains, how frequently it rains, what's going to happen to these crusts? And we've already seen some things that are kind of like the huge sandstorm in Phoenix. If you don't have these crusts, you're very prone to wind and sand erosion. And you may have also heard about these huge sandstorms in China as well. So trying to stabilize this crust and understand the climate regimes and areas that will be most susceptible to climate change is very important. And our project here at Berkeley Lab, like I told you, we go out to the field. Here's Eoin right here. Collect these samples, and then we bring them back in the laboratory, and we can subject them to different climate, wetting cycles, daylight cycles, temperatures, and study their metabolism in great detail. So here we can measure their respiration, we can measure their metabolism, and understand how do they accumulate carbon? Are there good periods, and then under what conditions do they start losing carbon into the atmosphere and maybe start to break down? And through this kind of study, we can develop a mechanistic understanding so we could predict and have models that could predict how climate change will affect different ecosystems, different desert crust ecosystems. And I wanted to close with this picture here. Here's Israel, here's Egypt, and there's this sort of bizarre line right here, and that's the border. Any guesses from the audience as to what's the difference between these two areas here? Why is Egypt white and Israel is this dark color? They're both deserts. Goats, very good. Yes, they graze over on this side. So these crusts are very robust in many ways, but they're not strong against being crushed. If you step on them, they turn to dust and they blow away. And they take hundreds of years to regrow. So being able to predict conditions where they may be sensitive or not is very important so that we can change our land use practices and keep that soil, that carbon in the ground. So thank you very much.
Okay, so we've heard about the tropics and about the deserts. Now we're going to go north to permafrost. So this is a picture from Alaska, and we're looking at a beautiful picture, actually, of permafrost. And permafrost is permanently frozen soil. So it's soil that's been frozen for thousands of years. And you can see here this dark purple region is the region in the northern hemisphere that is currently contained or underlain by permafrost soil. So it's a huge region of our hemisphere that is underlain by permafrost. Now why is this important? Why are we interested in permafrost? Well, this is another image of the carbon cycle, and we had a very good introduction to the carbon cycle from Margaret. And she explained all of these inputs and net and fluxes. But one thing that I want to focus on and she also mentioned soils. So soils contain a huge reservoir of carbon. So these are 2,000 gigatons of carbon that are currently contained in soils. Now, if we look at permafrost, the permafrost region has basically trapped a huge reservoir of that soil carbon. So of that soil carbon, a vast amount is trapped in permafrost. So about 960 gigatons of carbon are frozen in permafrost. Now, what happens as the climate warms? Well, frozen things thaw as things get warmer. So this includes permafrost. And as the permafrost thaws, that carbon can be decomposed by microorganisms and release greenhouse gases, CO2 and methane. So the permafrost is not so permanent. So it's really not permafrost anymore. And there are some signs that warming is already starting to impact permafrost in the Arctic. So what this photo shows here is what's called a drunken forest. So the permafrost underneath the trees has started to melt, and the trees are starting to topple over. So they look like they're drunken. And what this woman is doing here is she's lighting methane. So the permafrost has melted, and some pools of methane are bubbling up from the permafrost, and she can just light it with a match, and it gets a flame. So this is the cycle then, and Margaret also already introduced this, but for the Arctic, it's a huge problem because as the temperature rises, the permafrost thaws, you have the methane and CO2 released, more heat absorbed, and so you have this feedback cycle that is really aggravated. So why am I showing this? Well, I think that this is a very good illustration of what is in permafrost. So maybe you guys have heard about finding mammoths in permafrost. And I think this is the view that many of us are more familiar with. Well, it's not only mammoths that are frozen, it's microbes that are also permanently encased in this frozen soil. And so this shows these are green bacteria in soil. So we have mammoths, we have bacteria that are frozen in that environment. So what are they doing there? Well, as the permafrost starts to thaw, there's a lot of organic carbon that is frozen and stored there, but it becomes available to these microorganisms that are there. They wake up, so it's similar to the situation we had in the desert crust. These organisms, they're dormant, but as soon as it becomes possible for them to become more active, they can use that carbon and start to decompose it, and in the process release these greenhouse gases, CO2 and methane. So what we've done at Berkeley Lab is look at several different sites in Alaska, and these are some of the areas we've looked at in central Alaska, and they were chosen because they represent different kinds of permafrost. They mainly vary with the kind of plant material overlying or the amount of organic matter that is in the soil. And we have collaboration with the USGS, and so these collaborators go out and they take frozen cores of permafrost, and so we want to have it frozen and intact and transported to the laboratory. And then once it's in the laboratory, this is one of my postdocs, and she might even be here somewhere. This is Jenny Holtman. But what we want to do is ensure that our samples are not contaminated. So when they drill, we can maybe get a little contamination from the drill. So we take back a portion of the cores, these frozen cores, and while they're frozen, she sprays the surface of these cores with these fluorescent microscopic beads, and then saws off every surface of the core, and then checks in a microscope to see if she can see any of those beads. If she can't find the beads, then we're pretty sure that it's not contaminated, and then we go on. So this is the sort of tools that we're using, and many of us are using these tools. This is something the Berkeley Lab is kind of famous for, and it's called an omics pipeline. So we molecular microbial ecologists like to call things omics. And so you have genomics, proteomics, metabolomics, and I think this is nicely illustrated in this pipeline here. And depending on the omics tool that you're looking at, you get different kinds of information. So for example, if you're looking at the microbiome or microbiomics, you get information about the community composition, so which microorganisms are there. If we then go to the next step in the pipeline, this is metagenomics. Metagenomics is a tool that you use to sequence all the DNA. So you get your sample, extract DNA, and sequence it. And this is a protocol that Eoin was mentioning. This is called metagenomics, and that's nice because we get information not only about who's there, but what they're doing. You get their functional genes as well. However, that does not inform about whether the organisms are active. If we want to know whether these organisms are active, we can extract the RNA and sequence what we call the metatranscriptome. And this informs us about which genes are expressed, or the protein, that's the proteome, or metabolites, and this is Trent's specialty, to get information about metabolites. So this is just an example of some of our metagenomics studies, and each of these represents a gene, different kinds of genes, and these are different samples. And this would be a core. And what we see is that just by looking at the colors, if you compare the colors of these genes, so the green, they're less abundant, and blue, they're more abundant, you can see that they're different in the permafrost compared to the active layer samples. So there's some differences in the genes. So what is the active layer? I should define that. The active layer is the soil on top of the permafrost that is seasonally thawed. So it's called the active layer. So once a year it's thawed. whereas the permafrost does not thaw. So what we wanted to know was, first, which microorganisms were there in permafrost that had been frozen for thousands of years? That was our first question. And one thing that we found that's very interesting, especially in context of the former speakers, these microorganisms are actinobacteria, and two of our other speakers were talking about actinobacteria. So this is one type of actinobacteria, which is called Rubrobacter, and these bacteria are known to be very resistant to radiation and to desiccation. And so what we think is happening is that in this very old permafrost, that these organisms are surviving probably in brine films along the soil grains. We also find this organism here, so each of these bars represents a different organism, a different bacterium, but this one that we found was in every single permafrost sample that we've looked at. So we think it's very interesting. However, it's never been cultivated. We don't know anything about its properties. We do know that it's there. So the next aim would be to understand more about the physiology of that organism. One thing that we're very interested in is the methane cycle. And we know that different microorganisms function either to produce methane, so use CO2 and hydrogen to produce methane, whereas others use methane to produce CO2 and hydrogen. So you get a cycle here. So production of methane and using methane as a substrate to produce CO2. When we looked at our metagenome, so looking at all the DNA from frozen permafrost, we found something surprising. We found a newly discovered methane-producing microorganism. Now, this organism has never been cultivated, but when we looked at all of the DNA from the samples, it assembled into this nice draft genome, and we were very happy to see that. We didn't expect it. So this is a novel methanogen. Its closest relative is about 65% similar, so it's never been seen before, and it's never been cultivated. But because we could sequence all the DNA, we could discover this new organism that is probably responsible for production of methane in our samples. So based on this kind of data, all of these red lines here are supported by looking at DNA. So we just sequenced the DNA, and this allows us to understand something about the processes that are happening in permafrost and the active layer. For example, we know that the organic carbon is being degraded. We find genes for that pathway. We also find methane production and also methane consumption to CO2. We know that there's trapped methane that is also consumed. And so having this information helps us to fill in some of the gaps in these models so that we can get better estimations of the fate of this organic carbon. And so this is my last slide, and I wanted to bring us back to where we started with Margaret's talk, because really the aim of a lot of the work that we're doing at the Berkeley Lab is to be able to make better climate models and to get better predictive models. However, most climate models start at this end of the spectrum. You start with a satellite, top-down approach, and try to get information in that way. What we're doing is starting from the bottom up, where you have a lot of information about microorganisms and processes and environmental data, gas flux data. However, it's very hard to link these two types of data sets together. And so this is a huge focus now at the lab, is to try to get communication between the climate modelers and the people who are producing this very detailed molecular data to get better models. And so I think I'd conclude there.
Okay, I hope you'll all stay if you can, because we're going to have a question period. I was going to take some time to sort of get a dialogue going, but in light of the time and the fact that we have to be out of here by 9 o'clock, I think we'll go straight to the questions. And I just want to, if you sort of have a question, something puzzled you, there's no such thing as a foolish question. And if you don't understand something, I can guarantee there'll be other people in the audience who also don't, and they'll be very grateful if you ask the question. So if you could line up questioners at the microphones. Okay, let's start with you.
Okay, I want to start at the beginning. I really want to know what that wonderful imagery was at the very beginning. When we were waiting, that wonderful photography. I don't know. It was fascinating.
I shouldn't get any credit for that at all. I borrowed it from a colleague, John Crawford, and his student works with game theory. And so it was actually used from a soil grain that they had imaged, and then he had the tools, technical know-how to make it into this nice game-type image.
Okay, a question here.
I guess what I'm saying is I'm hoping that you are soon going to be able to drive that golden spike between the bottom-up and the top-down railroads, because every time I listen to statistics like 79% of the population in the US used to believe in climate change, and now only 59% does. I wonder if you have any insights into how we can seem to offer the kinds of sure answers that Fox News or Pat Robertson might offer. Because every time we say, well, we need to find more before we can really say anything, it seems like we lose a little bit of our credibility and our funding and everything else. It's good that Berkeley is doing this, but do we have any PR?
This is a question that I'm quite passionate about. It's a very important question. And one faces it all the time when one talks to the public about global warming. And the one thing that I emphasize more than anything else is that, yes, there are uncertainties in the projections of what future climate will look like. There's no question, but that our models are not precise. We can't make detailed, accurate, precise predictions. But what's really compelling from all of the science, including some of what you heard tonight, is that the uncertainties are skewed in the direction of the situation being worse than the IPCC projects, not better. If you look at one feedback after another that's not included in the models, these positive feedbacks that the speakers have mentioned, they are such that if we could include them in the model, if we had this science developed to the point where we could put them into the climate models, we know that the climate models would be making more dire projections, worse climate change, more hotter future, drier future than what they now project. So that is my answer to the skeptics at Fox News and the politics, is that yes, there are uncertainties, but if we knew how to narrow those uncertainties, it's not at all uncertain what the outcome would be. It would be more dire projections. Yeah. Does anyone want to add or subtract from what I said? No. Okay. Question here.
We learned tonight a lot about the carbon cycles. I wonder if there's been much thought given to how climate change might affect the oxygen cycle.
Okay. I think somebody want to deal? If not, I will. But
well, just in terms of the amount of of oxygen in the atmosphere, we're kind of lucky on this planet, the time that we live in, that there's about 20%. It's enough to breathe, not enough that we combust spontaneously. It's just right. But could happen. It would only take a few percentage more. So, but it's actually a lot of oxygen. So I may not be totally understanding your question, but if we were to combust even a lot more CO2, it would be a very small change in the amount of oxygen. All that oxygen gets consumed when you combust CO2, and this fossil fuel emissions would do it. will consume CO2, but it's a very small effect. Most of the carbon, the reason we have so much oxygen in the atmosphere is in paleo times, organic matter was buried. So we kind of disrupted this cycle of oxygen and CO2 trading off. So you've buried a lot of carbon that left this oxygen in the atmosphere, but most of that can't be burned. It's kerogen and carbonates in rock.
Yeah. If we could burn all the organic matter underground, the deep fossil fuels that are fortunately are inaccessible, we could use up all of the atmosphere's oxygen.
But the sort of known fossil fuel reserves, 500 billion tons, is not so much compared to all the oxygen.
It's a small fraction of a percent in oxygen terms.
I hope I said 5,000, not 500.
Yeah.
Okay.
This question, I believe, could be put to all of you, but I'm going to special to you, Margaret. How much cooperation or input is given to you by the agricultural industry or by the forestation industry, since I see that they probably have a lot in common with what you've given here tonight? Would you give comment to that, please?
Well, I will say that a lot of the collaborators that we have are supported by USDA, Department of Agriculture, and Forest Service. They're really concerned, but they're not just concerned, I think they're motivated because ecosystems can be so much part of the solution. So since terrestrial ecosystems are taking up carbon, they're moving it from the atmosphere, and we depend on them for so many services, they're really interested. In terms of industry side, maybe not as much, right? Just don't have as much interaction.
I was thinking particularly the private industry side.
Well, groups like Weyerhaeuser, the forestry industry is really interested, very much so. And in part that's business, because in terms of making biofuels, once you figure out these microbes that break down the tough material that makes wood stay upright year after year, but you get the microbes that can do that, what they grow, they say, hey, we could be growing wood, we can be growing biofuel, it doesn't matter to us. You tell us what the high value is, and we'll go there. So they're really quite interested in it, and they're happy for people to come do research in their watersheds.
Thank you.
Yeah.
So we have a question in the balcony over here, John, to your right, upper right.
Up.
Oh, yes.
I have a question that had to do with how the microflora in the soil affects human health. I have heard that our health and our intestinal health and overall health is affected because especially in countries that have used their soil for intensive agriculture over many years, like the European soils and especially the Indian soils, and new studies are coming out. And if you have any information or comment on that, there's some disease that have been particularly linked to the depletion.
Janet, maybe
or
Janet.
I can start to answer that. Maybe Eoin wants to jump in too. And the other side of my research works with the intestinal microbiome. And so there's a lot of intensive research in understanding which microorganisms, for example, live in our gut. And one thing that is maybe of some concern is the possibility for opportunistic pathogens from a soil environment to, for example, in a hospital environment. This is an example that I can think of. And where if you have especially compromised individuals, a normal soil organism that would normally be harmless could in fact colonize such persons. But usually this is not an issue because the soil microorganisms, we're exposed to them every day in our normal lives, and so we've built up an immunity to most of those. It's just if something gets off balance, then you can have an opportunistic, normally beneficial microorganism has the chance to survive.
Do you want to add?
Yeah, I can follow up on that. So there has been relationships drawn between dust events and increased asthmatic attacks, for example. And it's not just the particles, the soil particles, it's the biological particles. Outbreaks of meningitis have been associated with dust storms in Africa, and that's a bacterium, Neisseria meningitidis, that can be found in soil. But also, there's a hypothesis called the hygiene hypothesis. And this relates to people living in too clean conditions these days, over-sterilizing. And there's a direct connection between, I won't say cleanliness, but exposure to environmental allergens, dust, animals. People who grow up on farms are less likely to develop asthma, even if there's a genetic predisposition to asthma. So our exposure to the environment could potentially increase with climate change. The types of microbes in the air will change as drought changes. So that could have potentially important feedbacks to human health.
Yep. Thanks. So let's go here, and then back up to there.
Great. I'd like to see whether or not this rich panel can address what would be a kind of policy thought experiment, if you will. We here in our capacity to develop food locally and local healthy food throughout our nation have very compromised, historically robust biomes, traditionally called the plain states, that have now been totally monocropped, mined, and the communities, both our social communities and all the microbial communities, have been really, really wrecked. So the question would be, would there be a role in our upcoming Farm Bill, whether or not it be this one or the one 5 years later, where the business of kind of a ketoing subsidies towards, instead of getting rid of them, since it's our taxpayer dollar already, putting them towards investing or paying for fees for stewardship services. And among those stewardship services could be soil subsidies, where beginning farmers, existing farmers could then reoccupy these desolated areas, and Leopold Center or whoever through the land grant universities could basically begin to repopulate the appropriate balance of soil credits into those areas so that we could begin to heal our food systems. And so that's the question. So that's a thought experiment. So there you go.
I can start that.
Do you want to take that?
Sure, I'll start that one. There was a lot of different, in my mind, a lot of different points, good points, good questions about agriculture and the effect on the plains. And I do a lot of work in Oklahoma in sites that were affected by the Dust Bowl. There's very little topsoil, it's really poor soil, and I have a lot of good relationships with the farmers there, and they get government support to keep plowing and planting wheat, and it's a really hard job. When the climate's bad, they can't harvest. It's too wet, they can't get the tractor, and it's too dry, it dies, and they live a lot off of the insurance. But I think there's a lot of parts of the plains where we could help people move to, instead of annuals that you're planting every year and plowing every year and adding nitrogen every year, help them moving to perennial crops, which are much better for the soil and take a lot less input. But the farm extension services in those areas don't offer any input on that. They don't really help people go to no-till, and in fact, there's a little bit of pressure among the farmers not to do that. Some of the people that work for us have gone to no-till, and they're excited about their worms, but their neighbors are not. They just say, what are you doing, You got all those weeds, it looks ugly. It's kind of like almost a lawn thing. But it is an education thing, that people could get back to that if the Farm Service could, the farm extension, ag extension could move more in that direction. But in terms of regrowing on what you're calling the barren land, there already is a program, the Conservation Resource Program, the CRP land. This is land that's set aside, and it's really important habitat. And it's important not to think of that anymore as sort of abandoned or barren land. It's already being managed for wildlife, mainly, and for protection of streams in riparian zones. And I'm bringing it up because sometimes people will do projections of the amount of fuel we could get from biofuel production from abandoned land that wouldn't hurt cropland, but they're actually thinking about taking these reserves, Conservation Reserve Program land, taking it out of that and growing biofuels on it, which might be sort of grasses, not too bad, but it's harvested a lot. It's not the same. And I think we have to be really careful about, in my opinion, not trying to grow crops again on those lands, but we can take so much of the land that's already being tilled every year and changing the way that we're growing it. And that would be a change in both education and subsidy on those lands.
Yeah. Yes.
Yeah, I would just add something about not really farming, but grazing, that in the southwestern United States, that if we do have a value on carbon, there may be more value in leaving these very sensitive lands alone so that they can sequester carbon into the soil, as opposed to grazing a few head of cattle over these lands and destroying the biological soil crust.
How do you feel about planting, for either of you, planting trees on land that's no longer good for agriculture and doesn't have a lot of wildlife potential?
Is that relevant to?
Yeah, trees don't grow.
Not to your habitat, but I was thinking more to your habitat.
Yeah. Well,
to the lands that she's describing.
Right. So you might think, oh, we can plant forests in what used to be grassland. Is that what you're asking? Areas that used to be grassland. And that they definitely will take up carbon, and it looks really nice. But there's also some downside. That takes up a lot of water. When you plant trees where they haven't grown, you start depleting groundwater and start creating a little salinization of the soil. So it's, you kind of want to be good stewards. It's probably understanding what are the factors, the ecological factors that shape this landscape and make it what it is.
Yeah. Yep. Okay.
Hi, my name's Alex. I'm exploring the potential of biochar to improve the fertility of soils. And I'm curious about what the pathways are. Oh, one of the potentials or the exciting parts about biochar is that it lasts in the soil for a really long time. But when it does go away, how does it go away? What are the pathways by which carbon leaves the soil?
Why don't you start, Janet?
Maybe I can start with that. So we actually have an experiment going on at the moment where we're using biochar. We're all working together in this, where we're asking that exact question. How long does biochar last in soil? What are the rates of decomposition of biochar in different types of soils, including this tropical soil with the high rates of decomposition and some grasslands from Northern California? And permafrost. And permafrost, an active layer in other regions. And we're isolating microorganisms, we're identifying microorganisms. We've identified a group of microorganisms that can degrade biochar, and we're actively looking at those mechanisms. We don't know what mechanisms they're using right now, but they're likely the very similar mechanisms that microbes use to degrade structural components of plants, which is what biochar is. It's more aromatic, but the same enzymes, the same oxidative enzymes that the organisms use to break down lignin would likely be used to degrade biochar. And some of what we're seeing, those same organisms, for example, the actinobacteria that we're seeing degrading plant litter, seem to be also involved in degrading biochar. So we can answer some of your question right now, but this is ongoing work.
So part of that, and that really simple answer, would be that some of it is decomposed just like any other organic matter and leaves the soil as CO2 back to the atmosphere. But it also can be leached with water down into the profile. And also black carbon or biochar is very light. Sometimes it's sprinkled on the top of the soil. And then if you have a heavy rain, it just kind of floats in the runoff and just runs off, off the field. So it can leave in all three ways. So.
Anyone else want to add something on biochar? Okay. Up here.
My question is sort of building on the other prior two questions is what can you do to increase the flux of carbon into the soil in a way that doesn't disrupt our economy totally?
Are you thinking about agricultural practices or more generally?
I'm thinking, it's a general question.
You're very general.
Agriculture is obvious, but there might be other ones as well.
Okay.
Well, I'll just throw out something that I find pretty interesting. In China, they have huge problems with erosion. And now when they're building new highways, they're exploring the possibility of making artificial crusts, biological soil crusts along the side of the roads. And so it's a very active area of research in China, how you can stabilize soils. Like here, we tend to plant grasses and things like that along our highways. But in these very arid regions, like much of China, you know, the Gobi Desert, places like this, those grasses won't grow. And so they're experimenting with how you can make stable mixtures of microorganisms that you can spray on the side of the road and water them for a couple months and stabilize you know, and let these pioneers go. And
so you culture these in big vats in the lab and then spray them from the truck
spray them on the truck on the road. And so that's something that's carbon that was, you know, in the atmosphere, and as these guys grow, it goes into the soil. So I think it's a, you know, huge opportunity for us. And not only that, but you know, you prevent these dust storms, which is why they're doing it. I mean, if you've ever driven in a dust storm, you don't know what to do, because if you can't see what's in front of you, but you're pretty sure there's somebody behind you. So you don't want to accelerate, but you don't want to hit your brakes. And so it's a, you know, dangerous situation. And also we talked about with the asthma.
Yeah.
You know, I was at a conference and they said crust is dust. So when it gets trampled, it turns to dust, and that's what we're breathing in these.
Trampled or when off-road vehicles destroy the crust in the desert, which is a huge problem. Probably not in China.
Probably, yeah. Probably more hurting.
Yeah. Let's see. Over here.
There have been a lot of soils on the planet that have been badly degraded. In the tropics, there have been peat forests that have been burned. There's a lot of pastoral land that have been overgrazed, and you know, there have been topsoils lost through bad agricultural practices. Do we have any idea how much carbon could be put back in the soil if we were to manage all of those areas consciously?
Can you come up with an estimate for how much could be put back with the crust restoration?
Yeah, I mean, I've tried to, so it would be less than probably 1/10 of a gigaton.
Yeah.
I think, just to kind of scale it. So
yeah.
I mean, overall, the numbers for, if you just had all better agricultural practice, those numbers, I think they're a little inflated, but it might be 10% of anthropogenic emissions taken up every year. So a pretty large number, actually.
That's the number I see, massive reforestation programs could end up storing a lot of carbon, both in the trees, and trees produce what are called root exudates that pump carbon down into the soil, sometimes rather deeply. Another thing that has affected here in California the carbon in the soil is the invasion of annual European grasses that occurred about 100 years ago and replaced our native perennial grasses. One of my graduate students just finished her PhD thesis showing that in places where the perennial grasses, the natives, still exist, we have higher soil carbon levels. And what happens is the invasive annuals are not as productive. Their roots don't go as deep, so they're not getting the carbon as deep down into the soil, and so it decomposes faster. And the net effect is less carbon stored in our soils. You can see this effect out in the Marin headlands, for example. Managing the plants that live on the land is a huge part of this. And then agriculture has a potential maybe for up to sequestering up to 10% of annual emissions, although I think those numbers are quite inflated, actually. Yeah.
Hi, I'm
Where are we?
Oh.
Up here.
Oh, other way.
Oh, up there. You're
yes.
right in the lights for me.
I am a farmer on the central plains of Montana, and I'm both practicing organic and conventional agriculture. My question is, with organic agriculture, I am doing more tillage to the soil. I'm not using chemicals. But I'm also in my conventional ag, I'm using glyphosate. And all of middle America is using glyphosate right now. And my question is, what does glyphosate do to the microbes in the soil?
That's a good question.
And probably everyone can understand, but if you don't use herbicides, then in order to get rid of weeds for planting, you need to till. I mean, that's one way. But if you decide you don't want to till, then you're going to use the herbicides. You have this trade-off.
Well, All I know about glyphosate is it is degradable. Now, whether it would have an impact, and I'm sure that there have been studies on the impact, many studies on the impact of glyphosate on soil microbial communities. So, I just can't, say exactly, I would have to go look at the literature. But glyphosate is degradable. It probably has an impact on some members of the community. But I don't know if anybody knows more than that. No.
Good question.
Good question. Okay.
Hi, my name is Shael. I have two quick questions and a comment. One is what eats microbes? The second is when a tunnel is built, what happens to the structure of the soil? And my comment is just in regards to passing of Andy Rooney. I thought it was a genius thought that he had shared once, was that he thought the electoral college should really be comprised of real professors and scientists. So I just wanted to share that.
Well, I can take the first question. Maybe somebody else wants to take the second one. I mean, what eats microbes? Well, first of all, other microbes. They can feed on each other. There are some that are predators of other microbes. You have virus in soil, a large amount of virus that can predate. These are bacteriophage, is what they're called, that can infect and lyse bacteria. And you also have small little animals like protozoa, single-celled animals that can also predate on bacteria in soil. So it's a war zone in there, literally. And you usually see population fluxes like this. They grow and then something predates and they crash. So this is a typical cycle of population densities.
Just like human populations.
Yeah.
Right.
Do any of you want to
I didn't
deal with
understand.
The second question?
Yeah, it was hard to hear this last part of your question. I think all of us are a little unsure what you asked.
What happens to the soil structure when you build a tunnel or put down a pipeline like is proposed through the center of our country?
Yeah, I assume you're thinking about the pipeline from Alberta to the Gulf.
Just tunnels in general, because
yeah, that one's in all of our minds.
It kind of destroys the whole structure of soil.
Yeah. You can assume that anywhere where roads are being built or pipelines are being built over open spaces, that the soils are tramped, are compressed, that there's leakages in the process. Trucks leak oil, and basically you have what's going to be a degraded zone right in the neighborhood of that pipeline. The problem, of course, with the pipeline from Canada that people are really concerned about is, well, really two things. One is massive leaks that come from ruptures, which can happen, leading perhaps even to infiltration of toxics into the Ogallala aquifer, which would be a catastrophe. The other concerns with the pipeline have to do with the fact that it's bringing a vast amount of fossil fuel available for us to use or other people around the world, and that this will mean a huge new commitment to fossil fuel burning and therefore a huge delay in dealing with global warming. So that's more of my big concern with the pipeline would be that last effect. I think we're up to there now.
The tunnels in general.
Yeah.
I think it would depend how it's built. I mean, a tunnel need not disturb what's 10 meters above the tunnel on the land surface if it's done right.
I was going to say there are a few places where they have built tunnels for looking at roots. There's a few places where you can walk underground and see and they've got a glass wall to the side of the tunnels. These are for research. but really neat. Then you can watch the roots growing and worms tunneling and moving leaves down and just kind of watching through this window on the soil.
There are also
and it
caves.
seems not to hurt the soil.
Yeah, and caves that you can go in and see the tips of the roots coming through the ceiling of the cave. It's a neat way to see the underside of an ecosystem. Up here?
That kind of leads to my question, too, is that trying to think about how to explain this to a kid. And it sounded like in the desert, when you showed the pictures of the desert and it getting damp and basically things coming from cowering beneath the ultraviolet protection of the sand and then surfacing and I guess growing or you know, are there like biomes below us and are they identified? Are there habitats? And is the desert a very, very narrow habitat? And is the tropics a very, very thick habitat? And if so, how deep is this activity going?
That's a great question. Well, I'll start off with the first couple millimeters. So the desert crust is, you know, the microbes that I was first talking about are the cyanobacteria. So they're ones that can get access to sunlight. And the microcoleus is one that has this neat adaptation to where it can actually glide along these tubes when it gets wet and find its way up to the surface. And then when it's drying out, it scurries back down underneath the sand. There's other cyanobacteria that are absolutely at the top surface. And they survive by producing lots of sunscreen. Yeah. So they're black. So those pictures that I was showing of the dark crust, those were just loaded with all of these sunscreen pigments. But as you go deeper down, you start getting below where the light is. So you can imagine diving into the ocean as kind of like you're diving down into the soil. There's no light. And that's where the heterotrophs are existing. And they're living off of what is excreted by the cyanobacteria. So you're right, it's a whole little ecosystem. But this ecosystem may just be the top couple millimeters.
Well, go deeper. As you go a little bit deeper, microbes like to follow plant roots. And that's their source of carbon for the most part, or it's a source of freely available carbon. So the further the rooting structure goes down in the soil of plants, the microbes will follow that. And the microbes in the area around the plant root, that's called the rhizosphere, there's roughly about 10 times more microbes there. They're more active. Biogeochemical cycling is much more active near the plant roots. So to some extent, that the depth that microbes go is really related to where the resources are in soil. But we've studied microbes at 3 kilometers below the surface of the Earth. And there are self-contained ecosystems down there that are independent of photosynthetic energy. So we have pretty much microbes from the mantle to Trent's crust.
Wow. Yeah. Okay.
Hi, I was wondering if you know of any work on how increasing temperatures might affect abiotic chemical reaction kinetics, which could possibly change accessibility of carbon to microbes for degradation.
You're all looking
Well, there's been a lot of really good work on how higher temperatures will affect abiotic chemistry in the atmosphere. Smog formation, for example, and the rest of the chemistry that affects air pollution levels. And the calculations that have been done show that higher temperatures, of course, speed up reaction rates. That's a very general principle in chemistry. And the trend is going to be that the pollution levels are going to go up under higher temperatures in the air. But you're asking about soil chemistry and water chemistry, I presume. The temperature increase in the ocean is also speeding up certain chemical reactions. And one of the things that's a non-biological effect of higher ocean temperatures is that there's more carbon outgassing to the atmosphere. As ocean waters warm, they can hold less carbon dioxide. Just like a warm beer goes flat. If you take a cold beer out of the refrigerator and then leave the glass for an hour, it'll have less carbon dioxide. The flattening of the ocean is going to increase the amounts of carbon dioxide in the atmosphere. It's another one of these positive feedback effects that could be very important in the future. But for soil chemistry, I'm going to leave it to you guys.
So we've conferred. So There are a lot of important abiotic reactions, like organic material coating a mineral surface or aggregates forming, and it makes it very hard for microbes to get at the material. It's one of the reasons carbon persists for so long. But in our deep study, while you were asking your question, we don't know if there's very much research on it. I think it would be a great topic.
Right, yeah, that's exactly what I was asking. Thank you.
Okay.
We do have a colleague.
Question up there.
That's your dissertation. Okay.
Yeah, I just had a quick question. I guess this is more directed toward Janet. On one of your slides, you showed a nice cycle of methane in permafrost, how methane is being produced and consumed. And I just wanted you to talk about what kind of feedbacks would higher temperatures have on that cycle.
Oh, you asked the perfect question.
Yeah.
we just had a paper accepted today in Nature that addresses exactly that question. So if you take frozen permafrost and you heat it and it starts to thaw, what we saw in our experiments was that we saw a burst of methane initially, but we did our experiments in closed systems, and so we saw also consumption of the methane after a little bit longer incubation. So we had a little methane cycle in our system here, and we identified actually that graph I showed of that methanogen that produced the methane. We identified that, but also the bacteria that were responsible for consumption of the methane. And so in this example, heating can cause both a release of methane, and if the environmental conditions are just right, you can have some of it mitigated and consumed by methane-consuming bacteria.
So what will happen if you do that in the much larger but closed system called the Earth?
Well, I got asked that question by about 20 reporters today. And the thing is that so methane is a more potent greenhouse gas than CO2, yeah. And so it would be a good thing. It would be less bad if it's converted to CO2. CO2 is still bad, but I did mention that it depends so much on the context. You know, as you saw, the soil is a very heterogeneous environment. So if you have the conditions are just right, where you have the right moisture content, right organic matter content, and the right microorganisms there, then it could be possible to have some of the methane consumed before it's released to the atmosphere.
Yeah. Good.
Depends.
Here.
Yeah. I read in the paper a couple of days ago where the carbon dioxide increased in the air by 6%. It's one of the highest increases ever. Also, if I spoke for the soil, for the oceans, for the trees, for the plants, I'd be screaming at the human race every day. I know many environmentalists and myself, I'm an environmentalist, who feel that change is not happening fast enough to save ourselves. And I think your work is very important, and it deals a lot with the effects of the pollution. But again, if I spoke for the soil, for the oceans, for the trees, for the plants, I would say the most important thing is to challenge the cause of the pollution, which are corporations, banks, and politicians. And I would urge all of you to become part of the Occupy movement. We had 50,000 people in the streets last week. 10,000 people surrounded the White House yesterday to fight the tar sands. And the Vietnam War ended because of people in the streets. And we have you've heard this expression, if you're not part of the solution, you're part of the problem. Well, I feel that if we're not all out in the streets, we're not doing the best to save the soil. Thank you.
Thank you.
Hi, my name is Vincent, and I wanted to say thanks. I'm not a scientist, and so I appreciate you, what you said in the beginning, kind of building a bigger sense of ecology to understand some of these topics. I guess for me, it's been really interesting to hear about these marginal spaces as the places where this carbon is being sequestered. And I guess I've never thought about the crust I've walked on in the desert before, or how the permafrost really is counting for a lot. And I guess it's been partially answered, but like, yeah, I guess the question about farming, like how much is the agriculture that people do play up as a part of this because it sounds like the permafrost is soaking up quite a bit of it, and we are doing all this work out there, but it sounds like the net effect of all that agriculture that we do is relatively low, and I was interested to know, as kind of a piece of the pie,
Yep.
what is our activity?
I'll let them speak to agriculture, but I just want to say in response that the overwhelming cause of global warming is fossil fuel burning. Mm-hmm. And if we don't cut back on coal consumption and oil consumption and natural gas consumption, mm-hmm. We're not going to solve the problem, no matter what we do in the agricultural sphere.
Yeah.
But agriculture is very important, and it itself is a contributor because a lot of fossil fuel consumption goes into food production. So you're absolutely right about that. But when you look globally at the scope of the problem, it's fossil fuels is where it's at and where we have to make big changes.
I had another small question. I wanted to know if anybody had any idea about genetically modified organisms and if they were creating unique situations or unique, microbiotic life. Thank you.
My PhD thesis in the mid-'80s was to develop a method to detect genetically modified microorganisms in the environment. This is when the industries really thought that there was going to be a lot of release of genetically modified organisms into the environment. However, since then, this has not been the case, and this is largely, microbes, because of the regulations, the regulatory restrictions, especially in Europe. So really, it's a non-issue for microbes right now. It's just too much work to go through the regulatory system.
I think we don't have a question here, but a plea.
We're going to take 3 more questions, 1 up here and then 2 here, and then we'll close.
Oh, okay.
Yeah.
Good.
So 1 up there.
1 up there.
My name is Maria. I have a second question. I see that plastic use and deposits in our oceans are a big problem. And I wondered, because most of the plastic would actually end up either on the surface or buried somewhere, and how would that have an effect over time, especially the plastic that under UV breaks into tiny and microscopic particles?
Unfortunately, a lot of that plastic is ending up in the stomachs of albatross and fish and marine life. So it's a huge problem just from the viewpoint of biodiversity. But does somebody want to comment on the abiotic fate of the plastic and its relation to microbes?
There is microbial degradation. It's just the rate at which it happens is very slow, and that's why it ends up in other parts of the food chain. Yeah.
It's a huge hazard to marine life.
I wish we could say something more positive.
Everywhere we look, I just imagine, like in a billion years, the Grand Canyon will have the plastic cake layer.
Yeah.
Climate change with its direct effects, for example, in Texas or flooding in Pakistan, they would have a direct interest, Australia especially, with its drought.
Yeah.
Then you don't have to worry about whether it should be politicized or not. For example, CFCs were regulated by not politicizing it, but the urgency of it by the gentleman over here, you saw in the vote in the last election that could have changed back what Governor Schwarzenegger signed into law, but the vote was like 55 to 45%. So when to politicize and when not to would be a public relations matter. But that Koch-funded study by Richard Muller that showed that, in fact, climate change is real, global warming is real, was not publicized very much recently. And maybe when a skeptic funder funds a neutral observer and the neutral comes up with something to your credit or to this credit, it has more credence.
That's right.
but it's not publicized.
Yeah, it should have gotten more publicity. It got some. I think we have a last question here.
Yes, thank you. Thank you for the presentation. My question is, it's sort of a general question. At the end of the presentation, you mentioned how there's kind of a missing link between the bottom-down and top-up approaches to understanding ecology and climate, and especially soil ecology. And I'm wondering what you see as some of the most critical joiners of those two fields.
Okay.
Well, yeah, I can start. Some things that are missing out of current climate models or ecosystem biogeochemical models are explicit mechanisms of microorganisms representing microbes based on the fundamentals we understand of their metabolism and the way they perform biogeochemical reactions. And much of the reason for that is that these mechanisms weren't known, or they weren't very well understood. So as we gain more information on those, then finding ways of incorporating these into biogeochemical models at a lower level. So these are not global models, but biogeochemical models that are maybe ecosystem specific at a lower scale. And then using that information to better predict processes at that scale and then scaling up to global scales, I think is one way of incorporating this type of information and making it have an effect at a global scale.
Yep.
I'd just add that much of what we know about the biochemistry of microorganisms is based on organisms that can be cultured. And most soil microbes, or a large number of soil microbes, cannot be cultured. So it's just the void. We don't know. And yet these microbes aren't just a pure culture living in the soil. They're these complex communities of organisms. Just like, you look at a forest with trees and birds and all of the flora and fauna. That's what's in the soil. And it's how they're all interwoven that is the community itself. And so the desert crust to me is more like a skin on the desert. And it's very much alive and integrated. And yet we don't really know all the pieces of it to put it back together. So I think ultimately having some sort of model of how these microbes cooperate as a community is really critical. Like you started off with your talk, to understanding.
Yeah. In fact, Darwin, in my opening comments, I talked about Charles Darwin and how he was both the astute observer of the detail, that's the bottom-up level, the fossils and the detailed differences in the beaks of finches on the Galapagos. And then he looked at the sort of top-down scale. He didn't have satellites, but he could observe similarities and differences across continents. And these two scales came together and in his mind led to the creation of the theory of evolution. And I think that's I want to come back to my opening comments. I think that's the need, and what we're seeing here are pieces of this story beginning to be put together. But we're a long way still, I think, from putting the top down and the bottom up together into a coherent, grand unified theory of the biosphere.
So that concludes our evening. Thank you, scientists. Thank you, audience. We'll be back with Science at the Theater in the spring.